Hello everyone, myself Priyanshu Parakhiya and I'm a student pursuing Bachelor's in Engineering Physics at Indian Institute of Technology, Roorkee. In today's presentation, I'll be talking about magnetic resonance imaging technique and how we can leverage its principle for developing a method to image at nanoscale. So let's get going. The main theme of this presentation is going to be pushing limits of resolution up to nanoscale for the MRI methods. You might be having questions like what is MRI? Let's have a brief introduction to MRI. So in layman terms, MRI is like a super camera that helps doctors and scientists see inside the human body and other objects without needing to cut them open. It gives detailed 3D pictures of structures inside our body. MRI works by interacting with the magnetic properties of certain atoms, especially hydrogen atoms. These atoms act like tiny magnets and MRI can detect and manipulate them to create images. The regular MRI machines we often see have limitations in how small they can see. They can capture details down to the level of millimeters or smaller, but not as tiny as we might need for studying really small structures like protein and cellular structures. MRI has many advantages. It's safe because it doesn't have any harmful radiation. It can show different type of tissues with greater detail based on their magnetic properties, and it can even focus on specific atoms like hydrogen, carbon, or phosphorus. Achieving super high resolution is a big challenge though. We need to figure out how to get clearer pictures from very tiny volumes without getting overwhelmed by background noise. Now we'll be moving on to conventional MRI techniques and limitations. Conventional MRI relies on a method called inductive detection where cores surrounding the subject detect tiny signals emitted by the nuclei of atoms in response to a magnetic field. We use coils to capture signals from the body just like a camera captures light. But again, just as a camera has limits to how small it can capture details, so does MRI. Despite advancements, the smallest detail we can capture with MRI is still relatively large compared to tiny structures like individual cells or molecules. The highest resolution achievable today is around 40 cubic micrometers per voxel, where you can consider a voxel is like a 3D tiny pixel. However, there are some fundamental limitations. The first one is MRI struggles with small voxel volumes or smaller 3D pixels because the signal we get from the atom spins become weaker and weaker as the volume gets smaller. As we shrink the voxel size, the signal strength decreases rapidly, making it hard to detect and image small structures more accurately. These limitations mean we need new approaches to enhance the MRI resolution and image smaller objects, like pushing it down to the nanoscale. So to see things at the nanoscale, which is incredibly tiny, like you can think thousands of times smaller than the width of a human hair, we need new techniques that can pick up these faint signals and turn them into clear images, reducing the noise and increasing the image fidelity. So let's talk about the need for nanoscale MRI. The quest for nanoscale MRI arises from the desire to directly visualize and study the structures of biomolecules, and you can also consider nanostructures, which are crucial for understanding biological processes and developing advanced materials. Magnetic resonance force microscopy emerges as a groundbreaking technique offering unparalleled sensitivity, capable of detecting the magnetic signals from individual atoms and molecules. So let's dive deeper into the principles of MRFM. It contains three key steps. The first one is mechanical detection, where the MRFM detects tiny forces between the spins of atoms or molecules in the sample and a magnetic tip, which are generated when the spins align with an external magnetic field. The second step is spin manipulation, where radio frequency fields are used to manipulate the spins of atoms or molecules in the sample, allowing more control over their magnetic properties and their alignment with respect to each other. And then there's the third step where we use a 3D raster scanning method, in which a magnetic tip is raster scanned or passed over the entire sample, generating a spatial map of spin density. This process allows for the construction of detailed 3D images of the sample's magnetic properties. So now moving on. So let's consider an experimental setup for MRFM. Generally, in MRFM experiments, precise sample preparation and specialized equipments are essential. Let's consider an example setup where a sample comprises of tobacco mosaic virus particles or TMV particles immobilized on a cantilever-like structure, which is which you can consider as a tiny beam-like structure. And there's also a nanometer-sized magnetic tip mounted on a radio frequency microwire. This setup serves as a scanning probe. This complete experimental setup is contained in an environment which is maintained at a cryogenic temperature of around 300 millikelvin and in a vacuum to minimize interference from thermal noise and other environmental factors. To detect the tiny forces exerted on the cantilever, an interferometric method is employed which measures the oscillation of the cantilever with extreme sensitivity. So I'll be showing the experimental setup on the next page. So as you can see, this image illustrates the experimental setup used in magnetic resonance force microscopy for nanoscale MRI. The key components shown here are: the first one is an ultra-sensitive cantilever. This is a very sensitive mechanical resonator used to detect tiny forces. It has a sharp tip at one end. Then there's magnetic tip mounted on the cantilever. This is another nanoscale magnetic tip that generates a strong inhomogeneous magnetic field gradient. The sample under investigation in this case is tobacco mosaic virus particles deposited on the cantilever surface. Resonance slice. So as you can see, there is a thin, approximately hemispherical region around the magnetic tip where the magnetic field strength matches the resonance condition for nuclear magnetic resonance of the sample's nuclei. Then there is a microwire, which is basically a small copper wire located near the magnetic tip that generates the radio frequency magnetic field, which is the BRF, to excite the nuclear spins in the resonance slice. Then there is that IRF, which is a radio frequency current passed through the microwave, which generates the BRF field. Then there is a laser interferometer. So it is an optical interferometer which is used to detect minute oscillations of the cantilever caused by the magnetic forces between the resonant spins and the magnetic tip. Thus, magnetic tip is scanned in 3 dimensions relative to the sample, and the detected forces are used to reconstruct a 3D map of the nuclear spin density within the sample, providing nanometer scale resolution imaging capabilities. So what this setup does is it combines the principle of magnetic resonance imaging with the high spatial resolution and force sensitivity of scanning probe microscopy techniques. So now moving on. So let's talk about the resonant slides. As I said earlier, the concept of resonant slice plays a crucial role in imaging. This refers to a thin hemispherical region around the magnetic tip where the nuclear spins of atoms resonate with the external magnetic field. The thickness of this resonant slice is typically between 4 to 10 nanometers, dictated by a strong magnetic field gradient near the tip. Now moving on, the point spread function. The point spread function is a fundamental concept in imaging science, defining how a single point source in the sample spreads out in the final image. In MRFM, the PSF maps the signal bit from individual spin sources to in the sample. Calculating the PSF involves understanding the profile of the resonance slice and the magnetic field generated by the tip, both of which influence how the signal from the spins is detected and represented in the final image. So from the image here illustrates key aspects related to resonance slice and the point spread function in MRFM. The A part depicts the three-dimensional profile of the resonance slice, which is a hemispherical region again around the magnetic tip, where the magnetic field strength meets the resonant condition. The resonance slice is shown in red with P, the magnetic field of strength 2.697 Tesla, and the special coordinates X, Y, and Z are marked in nanometer. Then the B part shows the cross sections of the point spread function at different tip sample distances. This point spread function represents the response or signal contribution from a single nuclear spin at a given position relative to the magnetic tip. So moving on. Yeah, so as this quote states, a picture is worth 1,000 words. But in this case of MRFM, even a picture cannot provide proper depiction of the resonance slice and the point spread function. So to dive deeper into these topics and concepts, I'll also provide some resources link in the description. So now moving on to the next slide. So in MRFM, the raw data that we collected from the 3D raster scan doesn't directly represent the spin density of the sample. So instead, each data point is a combination of signals from various spatial locations. As I said, because of the point spread function, each spin emits some kind of signal which converges with the signal from other such spins, and which is why we need to convert this raw data into a meaningful 3D image. For this, we employ a process called reconstruction, which involves deconvolving deconvolving the acquired data with the point spread function. The PSF describes how the signal from each point in the sample spreads out in the final image. So the reconstruction process that we utilize is an iterative algorithm called the Landweber algorithm. So here's how it works. So the first step is initial guess. So we start with a initial guess of the spin density distribution within the sample, denoted as spin density not R. Then using this initial guess, we calculate a predicted signal map based on the convolution with the point spread function. So what we do is we try to relate how the initial guess differs from the predicted signal map based on the convolution with the point spread function, and then we compare this predicted signal map with the actual measured data and adjust our spin density distribution to minimize the error between them. Also, we to ensure a physical realism, we enforce a constraint that the spin density cannot be negative. And basically these steps are repeated iteratively until the reconstructed spin density converges to a stable solution. Now I'll be moving on to the next slide. So as you can see from the image, these are some results from the probing of the TMV virus particles. The first one, the A image, describes the three-dimensional reconstruction of the one hydrogen spin density for virus particles sitting on an adsorbed layer of hydrocarbons, whereas the B image represents a horizontal slice from the 3D reconstruction, showing the distribution of hydrogen in the plane located 13 or 14 nanometer above the hydrocarbon layer. As you can see, several virus particles are evident, and the C is the corresponding scanning electron micrograph. So as you can see, both are really uh, both are pretty accurate with each and matching with each other. So there is just some noise in the NRFM results, but apart from that, it basically represents the same. It basically represents the same slice. So in MRFM, groundbreaking achievements have been made in achieving remarkable 3D resolution, reaching down to the range of 4 to 10 nanometers for one hydrogen imaging of tobacco mosaic virus particles. This represents an astonishing improvement in volume resolution, exceeding conventional MRI capabilities by over 100 million times. It's like going from being able to see a large building to being able to distinguish individual bricks from afar. Importantly, the achieved resolution matches that of high-end electron microscopy techniques, which I already mentioned, that are traditionally used for ultra-high resolution imaging. This equivalence positions MRFM as a powerful and complementary imaging tool. MRFM's unique capabilities offer significant advantages over electron microscopy, including the avoidance of radiation damage and the ability to perform non-destructive 3D subsurface imaging. This makes it particularly valuable for studying delicate biological specimens, materials, and nanostructures. So let's moving on to the conclusion. Now talking about the potential applications. So why MRFM? The successful demonstration of 3D MRI using MRFM represents a groundbreaking achievement in imaging science. This marks the first time that such detailed imaging at the nanoscale has been achieved, opening new avenues for exploration and discovery of nanomaterials and biomolecules. This this milestone was made possible by the development of novel imaging methods and algorithms, high field gradients, and sensitive force detection mechanisms inherent to the MRFM. by overcoming the severe resolution limitations of conventional MRI. This microscopy method promises to revolutionize and provide unparalleled imaging capabilities, which leads to potential applications. Where the first one is structural biology, in which MRFM offers the potential for direct 3D imaging of protein structures and complexes, providing invaluable insights into the molecular machinery of life. Then there is polymers and materials science, in which MRFM can map chemical distributions within nanostructures, offering precise characterization and understanding of complex materials. Then the third is failure analysis. This is the ability to detect subsurface defects and contamination non-destructively, which makes MRFM invaluable for failure analysis in industries ranging from, let's say, semiconductor manufacturing to biomedical device development. And the fourth one is complementary imaging. So MRFM complements existing imaging techniques such as X-ray microscopy, electron microscopy, and scanning probe microscopy, offering unique advantages and enabling comprehensive multimodal imaging approaches. So this was so yeah, thank you for watching my presentation. I tried my best to possibly summarize MRFM imaging method, but because of its high complexity, you might want to dive deeper into its articles and research paper, which I have provided in the description. I apologize for any disruptions caused during the video. And again, thank you and have a great day.